Hi, my name's Kamlesh Patel. I'm a PhD student with the Physical Sciences for Health. I did my undergraduate degree here at the University of Birmingham in computer science with software engineering. I took a year to work and then decided I wanted to try a career in research. I applied for this CDT mainly because of the interdisciplinary nature of it. And with my PhD project, I've managed to work in computer science initially, and now I'm in the chemical engineering department and working with chemistry as well as the QE hospital. My project revolves around trying to create a sensor which detects sepsis. So sepsis is basically when you suffer from inflammation from a traumatic injury is my special focus, and the societal bacterial infection too. So you can suffer from inflammation and not have infection, in which case we don't need to administer antibiotics, which is a waste of money, bad for antibiotic resistance, and also destroys your natural bacteria, which prolongs your time for recovery, and it's just bad for you. So we want to try to target who to give antibiotics to. My project initially had two sections. So at the hospital already there was a big study called the Sc-Phy study where they collected lots of information from burns patients, included blood and urine samples, and they analysed those to see the metabolites or chemicals which are within those patients' blood and urine. I was tasked to analyse the data sets to try and find chemicals of interest which can be diagnostic for just sepsis. So we're hoping that the body reacts differently when there are pathogens there, such as bacteria, as opposed to when they're not present, and this response will let us differentiate sepsis and just generic inflammation. The machine learning took place in computer science. I tried different algorithms. So one of the issues with the data sets is that there's missing values and it's quite high dimensional for normal data. The missing values turned out to be a problem, so we tried techniques where we could build models which would allow us to impute data in, and so fill in the blanks in a slightly clever way. And we also looked at techniques which just completely ignored the missing values. This poses problems when comparing two samples, because if you're looking at a different set of chemicals across patients, it's hard to make comparisons between the two because you don't know if the missing ones contribute anything to the diagnostic properties. As it turns out, it was quite a struggle, and just due to the nature of the data set, we couldn't find too much of interest. The metabolites that we did find were just linked to metabolism in general, and so obviously if you become infected, your energy metabolism increases because your body's trying to fight an infection, and so those overshadowed any other possible biomarker. So because of that, we turned to literature, discovered some biomarkers or a biomarker of interest, and now we're trying to create a diagnostic sensor based around this. The group I work in do a lot of molecular imprinting, and so they use a technique called surface plasmon resonance to detect binding of specific metabolites, but we want to in the future, in order to make a bedside sort of clinical device, to move to fluorescence, because an SBR machine is quite big and you can't really move that around the university. And so fluorescence, you can get small readers and sometimes even a visual signal to show the diagnostic result. I've spent the last year and a bit now synthesizing a fluorescent chemical and running tests to do with the binding of the target metabolite, and also whether competitive metabolites will have the same effect. We're trying to create different techniques for getting quantitative data from this fluorescence, so how to measure the concentration in complicated solutions, so not just the pure solution but something like urine or serum, and then hopefully we'll see a difference when we come to testing clinical samples and how to progress this further. We can then reapply this with either the molecular imprinting and sort of fix it to a surface, but instead of needing the SPR technique we can just use the fluorescence itself, or other technologies.